All right. I ended up having to run. I should have said this before I started recording. I'm having a diverticulitis flare-up. You can ask about that some other time. If I end up having to run out of here, it's because of that. So, if we're willing, that might happen. But in fact, I'm thinking about it, means that maybe it could. So let's pray, and then we're going to start looking at some practical stuff of DNA and the life of a cell. Okay. Heavenly Father, would you help us today? Help us to be able to concentrate in cells and to appreciate how just the incredible order that you've put in place in our bodies controls so much, Lord, and how you control it. Father, I pray that you would just give us an awe of you. We ask this in Jesus' name. Okay. So we've talked about DNA quite a bit, right? We've talked about what it is, what it's made of, how it's arranged. We've talked about how it replicates itself. It can literally be self-replicating and largely without error. Your cells replicate themselves millions of times throughout your lifetime, and they do it by and large without mistakes. Now, mistakes can happen. You can get changes to your DNA. You can have changes to your DNA that influence changes to your cells. It can produce things like cancers, tumors, moles, stuff like that. If they don't, we probably understand pretty well at this point that your DNA regulates all your cell activity. Your DNA contains thousands of genes. These genes code for proteins, and your proteins do all the work. So if there's something that needs to be done, needs to be made, you need a protein for it. If you need that protein, you've got to go read your DNA. If there's a problem with your DNA, there'll be a problem with the protein, there'll be a problem with cell function, right? It's kind of like, I don't know if you've ever seen this, like a brick wall, okay? So, as long as the first course is laid straight, the whole thing will be straight. Everything will be nice and orderly. I saw an image once where they took a brick wall and they deliberately just put a book in it, okay? Just a regular old textbook. And they're just trying to, obviously their thing was like, hey, look at the influence that one book can have on things. This is a book that somebody had, and they laid it, and then they tried to lay a brick wall around it, right? And so as they're doing that, well, they're laying bricks, but then this course had to go up to accommodate for this book. And then all the rest of them could lay straight. Well, then what happened was it got more out of whack every course, because each time you build on it, that little bump from the book amplifies everything. Somewhat similar to the impact of DNA. If you get a mistake in your DNA, one little mistake, and A becomes a C, it has the chance to amplify, and literally that one little mistake can be the reason that somebody has a tumor. Somebody has cancer. DNA changed, which caused a protein to change, which caused a cell function to change, and now all of a sudden perhaps a cell that should have just been chill grows out of control, and it's not responding to the rest of your body going, hey, stop growing. And it just grows and grows and grows, and it takes resources, it takes up your blood, and that's how they find and stuff, right? They give you MRIs or CAT scans, and one of the things they're looking for in some of these is they're looking for blood flow, because a tumor, a cancer will attract more, the formation of new blood vessels, and it'll take more blood. So you just look at where the blood's going, and that's where you're going to have a tumor. By the way, this is how chemotherapy works also. One way you might think of a cancer, a tumor, is kind of like a vampire. They're just like literally trying to suck the life out of you. They're your cells, which is why your body's not good at destroying them. But they're just taking the nutrients that are like over-consuming nutrients, the blood. So if I begin to poison the blood, these ones that are over-consuming will over-consume that poison. And so in chemotherapy, they're able to, or at least they try to, poison the tumors And the hope is that you're killing them before you kill you. And so that's how chemo, and that's also why people get so sick and lose their hair and things like this. And that sounds horrible, right? And it is very, very difficult. But this is what your body does in so many ways. This is what a fever is. When you get an infection, a viral or bacterial, and you get a fever and it gets up to 101, 102, this is your body trying to kill this foreign thing before it kills itself. So you can't go around at 102 forever. That'll kill you. That'll denature, change the shape of your proteins, but your body's just hoping to kill them before it kills you. Okay? Well, that was a little side jaunt from where we're supposed to be. But the idea that DNA regulates cell activity, because that is what it does. It's so, so vital, so, so important. Well, I want to put into context DNA, its structure, its function, its activity with the life of a cell, with the life of you, okay? So, one of the things we're going to look at today is called the cell cycle. The cell cycle is it's the life of a cell. It's not the life of every cell that you have. You, by and large, have two types of cells. This is where now you really want to be writing this down, okay? Two types of cells. You have somatic cells, and you have germ cells. Somatic, soma. Have you ever heard this word soma? Okay. It means body. It's Greek for body. These are your body cells. All right, pencils down for a second. You'll come back to this in 1 second. Pencils down. Touch a somatic cell. Touch another somatic cell. Touch one. Bah, touch one. Touch another somatic. You're not touching somatic cells. These are your body cells. Okay. Most of the cells in your body that are yours, that's a whole other story, are somatic cells. Okay. Most of them are going to go through the cell cycle. You can go ahead and write now. Okay. Germ cells, on the other hand, they give rise to what we're going to call gametes, or sex cells. Basically, we're just saying eggs and sperm. So germ cells give rise to these. These are all the same thing, okay? These are not going through the regular cell cycle. These are. Okay? These are doing really some very different things than these are. These are simply replicating themselves over and over again. This is how one cell becomes 2, becomes 3, becomes 4, becomes 5, etc. Actually, I said that wrong. It's how 1 becomes 2, becomes 4, becomes 16. 16 times 16 is whatever that. All right. It's not 16 times. 16 times 2. But anyway, these things, they double in each generation, at least early on, after fertilization. So somatic cells, okay? You know what a cell looks like, right? What should I draw to draw a cell? Just big basics.
A circle.
Okay, draw a circle. What do you want inside your circle? Say another circle. Another circle. Somebody started to say nucleus, so that is my other circle, right? What's inside my nucleus?
DNA.
DNA. Good. Now, I'm going to draw DNA like this. Okay? Have I drawn DNA like this much for you before? Okay. Well, then I've been amiss. Okay? Inside of your nucleus right now, most of your cells will have their DNA looking like this. Okay? Do me a favor. Draw this for a second. Okay? You know that this stuff that looks like spaghetti or a ball of hair is DNA. But it's DNA in a particular state. This uncoiled state of DNA is called chromatin. Chromatin. Chroma is the Greek prefix. It means colored. The reason it's called this is that normally, if you just look in a cell, you can't see this. But if you put a stain in it, a colored stain in it, then this chromatin will stain. It'll become dark, and then you can see it. So most of the time, your cells exist as chromatin. Chromatin is useful. It's got all this genetic information. It'd be like having a series of books in your house, right? To be able to read a book, you don't want it covered in, like if you went to the store and you bought a book, or if you ordered it on Amazon, because of the age that we live in, and it came, how does it come? If it comes from Amazon, how does it come?
Packaged.
Packaged, right? And what do you have to do?
Unpackage it.
You gotta unpackage it, open it up. And maybe it has depending on what you bought, maybe it's in a box, or maybe I bought a Bible or two recently, so they come in a box, and then even when you open the box, they might have cellophane. Like, you gotta open that thing up if you're ever gonna read it, right? Well, this is open. This is accessible. This is unpackaged. But if you wanted to move, we are anticipating moving sometime in the next 6 months. Do I just want to grab every Bible, every book in my house, nice and open, and carry it with me one at a time?
That's sufficient for reading.
Huh?
You should. Sorry. No.
Okay. Wide open is a great idea for reading, bad idea for moving, right? What do I want to do if I'm going to move?
Package it.
Package it up in a box, right? Box them up, put them all together so they're not. And if I'm smart, I'm going to put like things together, right? Like I'm going to have some order to this, aren't I? Yeah. Have you ever moved? Okay. When you move, and I don't care how meticulous you are, this is something that happens. You're just sitting there, you're day 1, day 2 in your house, and you're like, where's the box with the shampoo? Where are the dishes? Because, when you move, you want to be orderly. Put like things together in the right box, label your boxes so you know where everything goes. Otherwise, it's a train wreck. And you might get to a new place. And what if you moved states? And what if something didn't make it, and that something was really important? What if you got there and your dad was like, oh honey, I'm sorry, I thought I put that in the Goodwill box. You gave it away? You gave away my shampoo? You gave away my best hairbrush, my makeup, my clothes? What? Why would you? I just made a mistake. I'm so sorry. Well, that doesn't do you any good, right? Like, it's gone. Well, what if that was genetic material? What if you made a mistake and you moved and you left genes behind that you needed? It'd be a big problem, wouldn't it? When your cells move, so to speak, when they divide, you need to make sure that each one gets a copy of every single gene that you've ever had. If they don't, there's going to be a problem at some point, okay? So, I've got this mess of chromatin. I have to have a way, this is every single gene you have. I have to have a way of, and this is fine for one cell, but when one cell wants to become 2, I have to have a means of copying every single gene that I have so that everybody has one, and I have to have a means of getting them to each cell without making a mistake. Okay? And so this is part of what happens with the cell cycle. So my chromatin, now this is the cell cycle, okay? So for the bulk of the time that your cell exists, between cell divisions, the DNA looks like this, okay? It just looks like this, and it looks like this, and it looks like this. It is chromatin, okay? Almost the whole time, because during this whole phase here, it's got to be able to read its DNA, or it's got to be able to copy its DNA. I guess this would be a good time to say, what does G1, S, and G2 stand for, okay? So G1 is the first growth phase. This is growth phase 1. So your cell, go like this. I told you before I'm big on hand signals and dances and stuff like that. Go like this. You'll be able to see. Okay, and it's got to grow. So have it grow. Have it get bigger. And stop. Okay? So that's growth phase 1. Then at the end of growth phase 1, we've got to get ready to replicate. Right now I have 1 copy of all my genes. I need 2 copies. And so I need to go into what's called synthesis. This is synthesis of DNA, okay, of DNA. That's what's happening in S phase. G1 is my first growth phase. Cell's just getting bigger. S phase, I'm doubling my DNA, replicating it. We did DNA replication before. That's what S phase is. And then G2 is my second growth phase 2. I'm going to draw these phases a little bit. Okay. Okay, so this is going to be G1, it's going to be S, it's going to be G2. Okay? To go from G1 to S, the cell has to grow, right? So in G1, I'm going to draw a couple, basically, S's to represent my chromatin. And then in S phase, I need to double that, right? And then in G2, I need to grow some more. And then down here, I have something called cytokinesis. Actually, let me do this. Let me draw this better. I like that better. That's going to be more helpful for you. What's happening during G1 phase?
It's growing.
It's growing. S phase?
Synthesis.
Synthesis of DNA, right? The DNA doubles. G2 phase?
It grows again.
It grows again. What's happening during cytokinesis?
It splits.
It splits. All right, time for hand signals. Ready? Go up. Little cell. Oh, just wait. We're going to do gang signs in a minute. You're gonna love it. Yeah. Gangster science, ready? All right, this gets bigger. G1 phase, get bigger. S phase, replicate. G2, and cytokinesis. Ready? G1, S, G2, cytokinesis. Got it? Okay, that's helpful. Now there's what's unique about this, because these are, they're the same size, they have the same amount of DNA, it doubled, we grew, we split, right? So this, it's a cycle, right? So we go from here, to here, to here, back to here, right? And that's the idea. So what's, like these look different, why? What's going on here? What do you see? What do you think it is?
There's no other circle.
Yeah, because during cytokinesis, the nucleus dissolves. It actually dissolves during the M phase, which we'll talk about in a minute. It dissolves. It's going to have to reform very quickly. It doesn't stay gone very long, but think about this. For this thing to split, like you've got to be able to get your DNA out, which your nucleus is in the way. So the nucleus goes away, and then it moves the DNA to the opposite sides, and all that kind of good stuff happens. What else is different? Look at your DNA here, and look at your DNA here. This is chroma 10. This is chroma somes. Good. So these are chromosomes. So how did we go from chroma 10 to chromosome? This is loose DNA. This is packaged DNA. Okay. This is loose on the shelf or on a desk. This is in a box. This can be read and used. This cannot. The only reason we do this is so that we can move the DNA. It doesn't stay like this very long. It's a very brief, because I need my DNA, right? So I can't just leave it in a box. I've got to package it so I don't mess this up, and then I've got to get it out so that I can use it. So this lasts very, very briefly, okay? This is a chromosome. Colored body. Like somatic cell. This is chromosome. And notice the nucleus is gone. So, yeah. And there's a step that happens in between. It's up there, it's called the M phase, right? Do you know what it stands for?
Metaphase.
Well, metaphase is in there. It's one of, metaphase is the M of the PMAP here. It's mitosis. So these three together are mitosis. Okay? Mitosis has 4 steps. This is where the gang signs come in. It's going to be great. I'm looking forward to this all year, haven't you, Kevin? You should have been. If you only knew, you would be. Mitosis. Mitosis could probably best be defined or described as the division of nuclear material, or division of genetic material. How one cell becomes 2, and each of those cells is sure to get a copy of all the DNA. Okay? So it has 4 phases. You have prophase, you have metaphase, anaphase, telophase. You realize as I did this, should not have had you draw chromosomes under cytokinesis because I'm going to contradict it in just a minute. Okay? If you want, you can loosen up, you can erase chromosome. I did that to point something out, and yet I inadvertently created an error, and I'm sorry for that. So this can go back to just loose chromatin. Okay? I do apologize. And for that matter, you better put your nucleus back. I know, I messed up. Sorry. That is my fault. I combined two things that I should not have. All right. In prophase. In prophase, two major things happen. I'm not going to make it, y'all. I'm in trouble. I'm going to put this on for you, and I will be back as quickly as I can. Let's see here.
Produce cells. So, one cell can copy itself to become two cells. The two daughter cells are identical to the original parent cell, exactly the same. Each one can complete a cell cycle to make copies of itself until you end up with lots of cells. This is needed for growth, and is how you went from being one single lonely cell to being made of trillions of cells. That's how you grew. We also need the cell cycle to replace worn out cells and heal tissue damage. So it is muy importante. Alright, so the cell cycle has three main parts: interphase, mitosis, and cytokinesis. Let's begin with interphase. Interphase is the part of the cell cycle where the cell is not divided. Think of this as normal cell life. The cell is doing normal cell things: growth, metabolism, and so on. Also, during interphase, you should notice that the nucleus is intact and that the cell's DNA is all stringy, like spaghetti. This stringy, uncoiled form of DNA is known as chromatin. There's one really important thing that the cell does during interphase that is needed in order to do mitosis, which is the next step. The cell replicates or copies its chromatin. Notice that each piece of chromatin is now paired up with its copy. We call these paired up copies sister chromatins. So, now the cell has copies of its DNA. This is critical so that the cell can divide up its DNA into two cells and still have them be identical to the original parent cell. One final thing I want to make clear. Interphase is not part of mitosis. It happens before mitosis and is especially important because it is where the cell's DNA is copied. Now, let's begin mitosis, the process of dividing the cell's DNA into two identical daughter cells. Mitosis has 4 phases: prophase, metaphase, anaphase, and telophase, which you can remember as PMAT. The first phase of mitosis is called prophase. During prophase, there are a few key events. 1. Chromatin coils and gets nicely packed to form chromosomes. So instead of being like a tangled mass of spaghetti, your DNA is tightly packaged and easier to organize and move around. 2. The nucleus goes away. It literally disintegrates. Why? Because it's in the way. The chromosomes are trapped inside the nucleus and can't get moved to where they need to go unless the cell gets rid of it. So it does. 3. The spindle apparatus, a bunch of protein tubes that are used to move your chromosomes around, also forms. It makes sense that all these things would happen during prophase, the beginning of mitosis, because they're the things that need to happen at the beginning. It's like moving to a new house. There are some things you do at the beginning of the process. Just like you need to pack up your stuff before moving to a new house, the cell needs to pack up its chromatin into easily movable chromosomes. And just like you need to leave your house and move out, the chromosomes need to get out of the nucleus, so it vanishes. Your whole house doesn't exactly vanish, but you get the idea. Finally, if you're going to move, it makes sense to have your moving truck ready to go. The moving truck is like the spindle apparatus. Again, it makes sense to have this at the beginning of mitosis. So, that's prophase. The rest phases will go much quicker. Metaphase is the next phase. In metaphase, the chromosomes line up down the middle of the cell. Think M for middle. The chromosomes line up down the middle. This makes the chromosomes easily organized so that they can be evenly divided in half in our next phase, anaphase. During anaphase, sister chromatids are separated from one another and pulled to opposite ends of the cell. Telophase is basically the reverse of prophase. Each newly formed daughter cell is going to start its new life in interphase. So we have to undo all the mitosis stuff that helped the cell to divide. Back to normal cell life. So, the chromosomes uncoiled form chromatin, the nucleus returns, and the spindle apparatus breaks down because it is no longer needed. Usually at the same time, the cell completely divides in half, a process called cytokinesis. The result is 2 genetically identical daughter cells. That is the entire cell cycle: phase, mitosis, and cytokinesis. If you enjoyed this video, please like it, subscribe, and check out the other free games, quizzes, and interactive learning experiences at ByMan Biology.
People love finding me, but me, I love finding the perfect gift. Like for my friend Brenda, who loves coffee, or for my little dog Wolf, who eats big.
Thank you. Gifts delivered on time or your money back. Etsy has it.
I'll take come back anytime soon.
What did he say was wrong?
I don't even know what he has.
right? Okay. Prophase. First step, right? This has to happen before your cells can divide. Otherwise, they will not each get a copy of the DNA. Every single gene that you have, every cell needs a copy of it, right? You don't want one cell to have 2 copies of it, and this cell to have none, or this cell's got 2 copies of these, and this cell has 2 copies of those, but that won't work. It's going to cause problems. Everybody's got to get a copy of everything. All of your DNA is already doubled here. By the way, these things, they're called chromosomes, right? A chromosome is made up of 2 sister chromatids. I'm going to talk about this more in a minute, but basically your one single strand of DNA here tightens up. It coils up to form a chromatid. This is just one strand of DNA. That's all it is, okay? Now it's double-sided, right? But it's one strand of DNA. What happens during S phase is you make a second copy of this, right? It's the same, it's identical. So this other side is called also a chromatid. A chromosome consists of 2 chromatids. And notice I've drawn them kind of separate here, just so you can see that they're 2 separate things, but they're attached. And that's because at this phase they are attached. Okay? These 2 chromatids attach at what's called a centromere. Centromere. Okay? So they attach together because in a minute, what we're going to do is we're going to separate them. We're going to make sure that each one gets a copy. Well, in order to do that, I got to put them together first so I can divide them later. I want to make sure, like I said before, that each cell gets one of these. I don't want one cell getting 2 of these and one getting none, and another cell getting 2 of this other one and this one getting none. Everybody needs one copy of everything. So I put my copies together so I can, this is like just keeping track of everything, okay? Being nice and orderly. So I put my 2 copies together, right? During prophase, chromosomes form and the nucleus or the nuclear envelope begins to disintegrate. Those are the 2 things that happen during that. Look at me. You must know this. This will be, hey, I'm gonna go, hey, first of all, know this. What are the stages of the cell cycle? Well, you're going to tell me G1, S, G2, mitosis, cytokinesis. 5 things, right? G1, S, G2, mitosis, cytokinesis. Those are the 5 steps of the cell cycle. You're going to have to be able to tell me G1 is growth. S is synthesis of DNA. It's where it doubles. G2 is more cell growth. Mitosis is where and then cytokinesis is where the cell contents get divided up. Okay? So you'll have to know that. I'll give you a second to write down whatever you want there. I'll even repeat it. You must know the 5 steps of the cell cycle. G1, growth. S, synthesis or doubling of DNA. G2, a second growth phase. M phase or mitosis. That's how the nucleus gets divided up. And then cytokinesis is how the cell gets divided up. Mitosis must happen before cytokinesis. Otherwise, your cells are going to have problems. Right now, we're focusing on the mitosis side of things. The first step of mitosis is prophase. The 2 things you must know about prophase are number 1, chromosomes form. And number 2, the nucleus dissolves. Okay. The next phase here is metaphase. And during metaphase, my chromosomes align along the center of this plate. Chromosomes a line across the middle. Okay? Anaphase. In anaphase, I forgot to draw stuff. Sorry, you better have this. Did I tell you what that was in the video?
There was the sister things.
These are my sister chromatids, right?
Yeah.
Did I tell you what this was?
Template.
Well, this is the envelope right here. It's dissolving. This is called a mitotic spindle. We'll watch this again in a minute. And we'll fill in a few things, okay? This is called, so here's prophase, metaphase, here's anaphase. In anaphase, my sister chromatids separate and they pull to opposite poles. It should be P-O-L-E-S, shouldn't it? Should. So prophase, these form. Metaphase, they move to the middle. Anaphase, the chromatids begin to separate. And during telophase, DNA loosens back again. And my envelope begins to reform. You waiting for this? You anticipating it?
Anticipating it.
The gang signs? You're not? You should be. This is one of the best games.
I don't typically go against that, so.
You are today. All right. Now we're cabin's ready. So there's one other generic label for this that we haven't covered yet. I better do it now. I'll do it to the inside. This is commonly called interphase. Interphase consists of G1S and G2. And then you go into mitosis and then cytokinesis. Interphase, between cell divisions. So, like almost all of a cell's life is spent in interphase, between cell divisions. What's it doing? Most of that time it's growing. A little bit of the time it's replicating DNA, but much of the time it's just growing, okay? By the way, it can grow, replicate its DNA, grow some more, and then just stop. Just chill and like never grow again or never divide again, depending. It has to have some kind of a signal to it that would tell it to replicate before it would go into mitosis and cytokinesis. unless it's cancer, in which case it's stopped listening to the cells around it and it's just doing its own thing. And that's a real problem, okay? And this would be called a GO phase, where it's like we're done growing, we're just chilling, okay? But this is interphase. So, during interphase, during all of interphase, your DNA is loose. Loose DNA is called? Chromatin. Loose DNA looks like this. Go. Come on. It's just loose. Wiggle your fingers, wiggle your hands. But when you want to go into mitosis, the first step of mitosis is prophase. And then you have to form chromosomes. Form your chromosomes. Do it now. This will be the most helpful thing to you if you'll I told you it was going to be fun. This will be the most helpful thing to you. So interphase, prophase. Not bad. But don't put them in the middle. That's going to confuse you. Do them like, yeah, like real hood rat, all right? Ready? If you knew where I grew up, you'd be like, oh, he can say that. Interphase, prophase, metaphase, they move to the middle. Kevin's into this. All right, ready? Interphase, prophase, metaphase. Anaphase, we separate our sister chromatids and look at what it looks like. So we go like this. Okay. Interphase. That's good. Yeah. Interphase. Prophase. Metaphase. Anaphase. One more time. I'm saving the best for last. You ready? He's going to die in a minute. Interphase. Prophase. Metaphase. Anaphase. What's the next step? Telophase. What happens to the DNA? It loosens. No. It's better than that. Ready? Ready? Okay, wait for it. Ready? Interphase. Prophase. Metaphase. Anaphase. Telophase. You won't forget that, will you? I hope not. Because you need to remember it. Because if you just think about it, this reminds, interphase, the DNA is loose. Prophase, it tightens up, it forms into chromosomes. Metaphase, they move to the middle. Anaphase, they begin to separate. Telophase, they loosen back up. So it's not just silly and goofy stuff. This kinetic thing, this movement thing that you're doing should help you to memorize it. Okay? So interphase, prophase, metaphase, anaphase, telophase.
We did some like freshman biology. It was like metaphase, anaphase, telophase. We used to do this too in biology.
It was not as gangster.
Yeah. My teacher was not.
Not very gangster?
No.
Alright. No. Did you go to Fairfield High School?
Mm-hmm.
There you go. Alright. Prophase. Huh?
It's getting there.
Can't be that bad here. We can compare stories.
It's gone way worse.
If you haven't seen weave ripped out on the stairwell,
Oh
and
yeah,
afraid that people are about to die.
Like 7th
We're
grade.
going to not talk about this right now. Prophase, metaphase, anaphase, telophase. These are the steps of mitosis right after this. So this is how the nucleus divides, right? But then right after this, the whole cell divides. Okay? So there's a brief moment in time when a regular somatic cell has 2 nuclei in it, and then immediately the cell is going to divide following it, okay? This is how, this mitosis thing is how one cell becomes 2 with a complete copy of all your DNA. Again, if this goes wrong, your cell's in trouble. Your best case scenario in this point is that the cell just dies. Sometimes they don't. Sometimes they just go crazy. Okay? All right. Let's talk a little more. You feel good about this? Okay. Let's talk about your cells and your chromosomes and your chromatin and your chromatids. your cells have chromosomes? Do you know how many you have?
23.
Not quite.
23 pairs.
23 pairs. You have 46 chromosomes. Wait, before I even start that, let me say that's your somatic cells. What does somatic mean?
Body.
Body cells. Your somatic cells have 46 chromosomes. And as she pointed out, there are 23 pairs of homologous chromosomes. Means that for every gene, you have two copies. One from mom, one from dad. This means that every time that your body's like, hey, I need this protein, your cell goes and accesses two different chromosomes that are essentially the same. They have the same genes on them, but the genes might be in different forms. And so the proteins they make, it might make two copies of identical proteins, or it might make two copies of proteins that are very similar, but slightly different. Does that make sense?
Mm-hmm.
So at any given time, how many strands of loose DNA are in any one of your cells in M1 phase?
Forty-six.
46. Oh, I said M1. I should have said G1. I don't know what I was. I'm like, what did I say? In G1 phase, you have 46 strands of loose DNA. Makes sense, right? 23 of one of this, and 23 of that, 23 from mom, 23 from dad. But I want to divide this cell, so I need to get all 46 strands, a copy to this cell and a copy to that cell, the two daughter cells, right? They each need their own copy. I only have one copy of each one, so I need my 46 strands of DNA to become?
23.
No. That would divide it in half. That'd
Well,
be a
it's
problem.
46, whatever that is.
Yeah, so 46 times 2 is 92. So in S phase, 46 becomes 92. Six becomes 92 strands of DNA. In G1 phase, or I'm sorry, in G2 phase, I have 92 strands of loose DNA. What's loose DNA called?
Chromosome
Chromatin.
10.
In mitosis, I don't have loose DNA anymore. What do I have?
Chromosomes.
What are chromosomes made of?
Chromatid.
Chromatids. How many chromatids do I have?
We'll have 46 then.
No. A chromatid is made up of one strand of DNA. How many loose strands of DNA do I have?
92.
92. So I have 92 chromatids. And 92 chromatids equals how many chromosomes?
46.
46. Each chromosome has 2 sister chromatids. I'm going to erase a little bit over here real quick so I have more room. Then, after cytokinesis, how many chromatids worth of DNA does each cell have?
I'll go back to 92. I'll go back to 92.
Well, I had 92.
So you have 46?
I'll go back to 46.
Okay.
Now, notice how I wrote this. I'll have 2 cells with 46 chromatids worth of DNA. It won't be chromatids though, right?
Mhm.
It'll be chroma 10. It'll be 46 loose strands. Why does it need to be loose?
Because it's starting over, you have a nucleus again.
Say that one more time.
It's starting back over and you have your nucleus still there.
Yeah, but why is it, so yes, that is all true. And why does it need to be loose though?
So you can read it?
So I can read it. I can't read a book if it's in a box, right? Okay. This is good. This is very good. We're time. I got 20 minutes. We're not doing bad at all. Okay. 46 chromosomes. That's what all your body's cells have, right? Each chromosome, you have 2 copies of each type of chromosome. Your chromosomes are typically numbered 1 through 23. And number 23 are always your sex chromosomes, your X and your Y. Ladies, you have 2 X. Gentlemen, we have 1 X, 1 Y. Where did we get the Y from? Mm-mm. Your mom would be your dad if that were true. You got it from your dad. Where'd you get your X chromosome from? Your mom. Where'd you get your 2 X's from?
No.
No.
Oh, my dad.
Hmm? Where'd you get your X chromosome from? Your mom. Where'd you get your one of your X chromosomes from?
No,
Your mom. Where'd you get your other one from?
Dad.
Your dad. Your dad has an X and a Y. You have an X and a Y. I have an X and a Y. You have 2 X's. The difference between man and female is an X and a Y. That 1 chromosome difference is the only difference between being a male and being a female. Okay? And so whether or not you become a male or you become a female depends on whether your dad threw you an X or he threw you a Y. Make sense? Okay. But you have 2 of each type of chromosome. So you have 2 copies of each gene. One that came from mom, one that came from dad. And they might be, in many cases, they're the exact same. They're going to go to the exact same protein. Sometimes they're not, though. And this is the foundation of genetic variation. So I have 4 children. 3 of them have brown eyes. 1 of them has blue eyes.
Recessive gene.
It's a recessive gene. Which one? Do you know?
The blue
The blue eyes is a recessive gene. I have brown eyes. How does one of my sons have blue eyes?
The mom. No.
Well, all of them got the blue from mama. That's all mama has. My wife has blue eyes. So all my wife could give every single kid is blue. I gave 3 of the kids brown, which I got from my dad, by the way. My dad has brown eyes. And I gave blue to my oldest boy. And I have brown eyes. That means that one of the copies on one of the chromosomes, the homologous chromosomes, is brown, and the other copy is blue. They're different. Now, you only see brown. That's another thing that we'll get into later, probably not this semester because we don't have much time left, but we'll see. As far as dominant and recessive traits or dominant and recessive alleles. But I want to connect some of these concepts to things that are very practical. This is not just theory. This really matters for who you are and why you're similar to your brothers and sisters, but you're different. Why you kind of look like your mom or you kind of look like your dad, but you're different. Or why you have traits from each one. All your body cells have 46 chromosomes, 23 pairs. 2 copies of each gene, 1 from mom, 1 from dad. At G1 phase, you have 46 strands of loose DNA, but to replicate your cell, every cell needs a copy. So 46 becomes 92, still 92 in the second growth phase. Then in mitosis, we're going to work on taking that 92 and turn it back into 46. So those 92 chromatids form into 46 chromosomes. How many chromatids per chromosome? Say 2. 2. It's 2 chromatids per chromosome. They're connected at the centromere. Remember we drew them like this. This is what a chromosome looks like. Connected right here. So this is 1 copy of DNA, this is another copy of DNA. What's the relationship between the two DNAs here? They should be identical. Because here's how it worked. Here was my DNA, right? And then I opened it up, and then I copied it, right? During DNA replication. Do you remember that? I got Okazaki fragments and all that kind of stuff. Which I think it went the wrong way, but anyway. So this becomes one of these, and this becomes one of those. When the DNA replicated, when it opened up, and we had primase, and we went helicase, RNA primase, DNA polymerase, and then ligase. And one strand of DNA became, or 1 copy of DNA became 2 copies of DNA. Well, one of these is this chromatid, one of those is that chromatid. They should be identical. That's why they're called sister chromatids, okay? This, by the way, this happened during which phase of the cell cycle? Where'd your DNA get copied?
Oh,
S phase. So this that we did, a week and a half ago is S phase, this DNA replication. Okay. All right. So this becomes 1 chromatid. This becomes another chromatid. They join together. They're a chromosome. You have 46 worth of these. Okay. All right. Mitosis. We take those 46. Or these 92 chromatids, and we separate them, right? This should have said chromatids also. Well, no, that's right. Okay. After kinesis, you're going to have with 2 cells with 46 chromatids worth of DNA, 46 loose strands of DNA. Do you have any questions at this moment? I feel like this is a lot, and yet I have more to give you. Around you. You're saying no, I don't have questions, or no, don't give us any more.
No, I have questions.
Okay. All right.
I vote for no, don't give us any more as well.
My dear, we're already so behind. Now, Now let's talk about gametes. Gametes, so we've been talking about body cells so far, right? Gametes are sex cells, right? Gametogenesis. I can't spell, I'm spelling gametophenesis. Gametogenesis equals the formation of sex cells via, I'm going to say, mitosis. Another way of saying this is it's how we go from 46 chromosomes to 23 chromosomes. Another way of looking at this is from 92 chromatids to 23 chromatids. Make you pause and think there in a second. Kevin, how many chromosomes do you have?
46.
How'd you get them?
I was 30 from my mom,
23 from mom and
23 from dad.
23 from dad. If you'd have got a different number than that, let me say that's number one, you'd probably never have developed at all. Most miscarriages, or there are many, many times a woman may become pregnant very, very briefly, and it's a miscarriage and she may not even know it. Or it may develop, the child may develop a little while, and then you know, there may be a miscarriage pretty early. Sometimes it'll happen later. Almost always, it's because of a genetic anomaly. And most of those, especially the early ones, it's because of the wrong number of chromosomes. Something went wrong in the process, and that baby does not have 46 chromosomes. Maybe it has 44, 45, 47, 48, 49, some other number than 46. You cannot survive. You need 2 copies of every single gene. Okay? There are certain traits, certain cases where a child can exist with an abnormal number of chromosomes, but it's very rare. Okay? If you have only one of a chromosome, it's called monosomy. It means you'd have 45 chromosomes instead of 46. If you had 3 of it, so you have 47 total, that's called trisomy or trisomy. There are very few of those where a person can exist and develop. In fact, even if you were just missing like a major, or like a chunk of chromosome, you're missing enough genes for the baby to not develop and to not develop properly. So it's absolutely vital that your body, in its gametes and its sex cells, is able to take it from 46 chromosomes down to 23 chromosomes. Okay? Because that child has to get 1 copy of each gene from mom, 1 copy of each gene from dad. Now, right here I said, hey, 46 chromosomes, 92 chromatids. We're used to that, right? Because you think of a chromosome, and it's got 2 chromatids, right? And at the end of mitosis, I go from 96 chromatids down to 46 chromatids. But here I'm going from 92 chromatids down to 23 chromatids. Well, 46 is half of 92. And we know that, hey, we doubled our DNA and then we divided it. Everybody got one copy of everything. But I don't want my child getting one copy of every gene that I have, because then I'd be giving him 46 different chromosomes. I only want to give him 23. I need to give half. So I can't give him 40, or her, I have one daughter, 46 chromatids. I have to give 23. So I don't have one division of my nucleus here, I have two. This doesn't produce one cell, this produces 4 cells, okay? So let me draw that also. We go from this to this to this. And if you're a lady, that's what they look like, and if you're a man, then this is what they look like, right? The process is essentially the same. This would have 92 chromatids. This would have 46 chromatids. These would have 23 chromatids. This process is called meiosis. So mitosis is how we divided somatic cells to reproduce them exactly. Meiosis is how we make sex cells, and we don't want to produce them identically. Right? We want to divide the genetic material into 4. We want it to be half of what an organism needs. There are 2 cycles of meiosis. There's meiosis 1 and meiosis 2. In meiosis 1, I go from 1 cell down to 2. And in meiosis 2, I go from 2 cells down to 4. Stop there for today. I'll get into the details of all this. So this is kind of the big picture overview. We'll get into the details of the steps next time. One thing that's going to be nice is if you remember your gangster science, your prophase, metaphase, anaphase, telophase, we see those again here, okay? So it'll be helpful. Have a blessed day.
Thank you.